A pharmaceutical cleanroom can be scrubbed exclusively with high-end EPA-approved sterilants and still fail a federal audit. Inspectors routinely issue FDA 483 citations for lacking sanitizer efficacy studies. An FDA 483 is an official form used by investigators to document conditions that may constitute violations of the Food, Drug, and Cosmetic Act. This chart illustrates the mathematical requirements for microbial control in the industry. Sanitization requires a 3-log reduction in bioburden. Disinfection demands a 5-log reduction. To achieve a biocidal or sporicidal state, you must prove a 6-log reduction. A chemical manufacturer's EPA-approved label claim is based on highly standardized laboratory testing. It guarantees baseline efficacy under controlled conditions, which may differ significantly from the performance on a specific manufacturing floor. True pharmaceutical validation requires a facility to engineer a bespoke, site-specific testing pipeline to prove the chemical actually works in their specific environment. Rigorous, facility-specific empirical data provides the only mathematical proof of regulatory compliance. For decades, the industry has practiced disinfectant rotation, switching between different chemical classes to supposedly prevent microbial resistance. True antibiotic resistance is biological. It is mediated by plasmids, extrachromosomal DNA molecules that allow bacteria to exchange genetic survival traits with one another. Chemical disinfectants do not interact with genetics. They cause total, brute force physical destruction, denaturing proteins and dissolving lipids until the cell wall ruptures entirely. Microorganisms do not develop genetic resistance to this kind of physical obliteration. The pharmaceutical industry rotates disinfectants due to pressure from FDA inspectors, rather than any scientific evidence of chemical resistance. Understanding that chemicals kill through physical destruction, rather than genetic interaction, is the starting point for designing a valid testing pipeline. To understand how to test a chemical locally, we have to look at how it is tested nationally. The Association of Official Analytical Chemists, or AOAC, dictates the Hart surface carrier test required for EPA chemical approval. The standard preparation workflow involves cultivating ATCC strains and applying them to small physical carriers. ATCC strains are standardized, highly characterized microorganisms acquired from a global biological resource center. During execution, inoculated carriers are exposed to the disinfectant in a strictly controlled 20 degrees Celsius water bath. They are then transstored into neutralizing broth at precise 30-second intervals to ensure exact 10-minute exposures before incubating for 48 to 54 hours. The test is highly structured, but it contains a flaw that renders it insufficient for pharmaceutical applications: the physical composition of the carriers. The AOAC methodology relies on highly porous borosilicate glass or porcelain penicillinders. You will rarely, if ever, find these materials on a modern production floor. The baseline AOAC test proves a chemical kills bacteria on porcelain in a highly controlled laboratory. It provides zero data on how that chemical interacts with the specific steel alloys, epoxies, and polymers in your cleanroom. Bridging the gap between the lab and the cleanroom requires custom in vitro testing, utilizing physical samples of the facility's actual infrastructure. We replace the porcelain cylinder with a coupon. This is a 2 by 2 inch physical sample of the exact materials used in the facility, whether that is 316L stainless steel, vinyl flooring, or painted drywall. These coupons undergo a dual inoculation challenge. They are tested against the standard ATCC strains, but more importantly, against environmental isolates. Environmental isolates are the specific native strains of bacteria and fungi swabbed directly from your actual cleanrooms. Must be run alongside mandatory positive controls to prove viability and negative controls to ensure sterility. The test must also replicate the exact physical application mode used by the cleaning staff. Mechanical action, like wiping or scrubbing with a saturated cloth, physically shears bioburden off a surface, whereas passive application methods like fogging rely entirely on chemical contact. Contact times are then bracketed. Analysts test the chemical at multiple empirical intervals, such as 5, 12, and 15 minutes, framing the manufacturer's recommended 10-minute mark to find the optimum local variable. This data table shows the ultimate metric of a custom coupon test: By mapping a quaternary ammonium compound against stainless steel across various contact times, the laboratory can mathematically track and prove the required 5-log reduction. Custom coupon testing converts generic chemical claims into mathematically proven, facility-specific efficacy data. Before those coupon tests can be trusted, the lab must overcome the most complex technical hurdle in the pipeline, method validation, governed by USP Chapter 1227. USP Chapter 1227 provides the official guidelines for validating the recovery of microorganisms from pharmacopoeial articles and surfaces. Method validation presents a difficult paradox. To definitively prove your disinfectant killed the bacteria on a surface, your sampling method must be perfectly optimized to find and grow any bacteria that merely survived the attack. The primary threat is the disinfectant itself. When a contact plate touches the steel coupon, it absorbs residual active chemical directly from the surface. If that chemical residue is not immediately neutralized, it will continue attacking and killing injured bacterial cells while they sit inside the incubator. This yields empty agar plates, creating a false negative compliance report. The solution is chemical neutralization. Specific neutralizing agents perfectly paired with the biocide class must be integrated directly into the testing agar or transport buffer. This matrix shows the precise chemical pairings required. Quaternary ammonium compounds demand lecithin and polysorbate. Halogens, like chlorine and iodine, must be neutralized with sodium thiosulfate. Even with perfect chemical pairing, hitting the required 70, the factory expiration date is void the moment that concentrate is mixed with USP purified water on site. The resulting U solution is an entirely new solution with unknown stability. This visualization compares the efficacy of a 500 part per million sodium hypochlorite solution over time. The freshly prepared 0-day solution achieves complete kill. By day 60, the degraded solution exhibits a catastrophic failure rate, with 60 out of 60 carriers failing against Pseudomonas aeruginosa. Facility labs must empirically test their mixed-use solutions at multiple time points to define their own specific degradation timelines. Once the expiration timeline is set, the laboratory moves to the final phase of the validation pipeline, in situ testing. In situ tests are conducted in the real world. Technicians perform bioburden monitoring on actual production equipment before and after the cleaning protocol is executed. No artificial laboratory inoculation is used. This measures the protocol against the actual native flora of the facility. A compliant facility writes detailed raw material SOPs, tests customized structural coupons, validates recovery methods with neutralizers, determines use dilution expiries, and proves the entire system with in situ environmental data. Cleanroom disinfection functions as a continuously monitored, mathematically derived system engineered for a specific facility.